I leave you the stage, Max.
All right, thank you. Hi, everyone. So I'm going to start right away. So the most common approach in the field right now to characterize brain damage in MS is looking at lesion load. And this is a structural index derived from structural MRI. And it is often reported as a simple volume, actually, the volume of the lesions in the brain. And I was quite surprised to see that actually only 5% of the studies do report it as a ratio, so as a normalized volume of the lesions over the brain tissue volume. So for the rest of the presentation, I will, when we talk about lesion load, I'm going to talk about the normalized version, which makes a bit more sense than simply reporting a volume. So the problem with lesion load is that it correlates only modestly with clinical assessments, and this is called the so-called clinical radiological paradox. And the reason why is quite simple, actually. Lesion load, as you might see, is agnostic to location of lesions in the brain, meaning that if you have a lesion in an eloquent area, it might be more damaging than one that is not located in such an important area in the brain. And the other reason is, of course, microstructural damage is poorly visualized by conventional T2 image, for example, or T1. So as we know, of course, diffusion MRI has looked into providing another set of metrics that will go beyond lesion load, but the relationships with the clinical scores do not always exceed or do perform better than a conventional lesion load approach. And the reason for that is also quite simple. First, if you look at studies that look at tracks in MS, there is a premature termination of streamlines within the lesion, so even before reaching the lesion. So if your whole analysis is trying to analyze the effect of a lesion on a tract and you're not even tracking through it, there's a problem already at this stage. And of course, the ambiguity associated with the DTI-based metrics that we use. We all know the limitations of, for example, FA to characterize microstructure. So in recent years, there's been advances in local modeling and tractography, and we are now able to track more reliably inside lesions. And when I say now, I mean really in the last 2 years. As you can see here, there's a couple of studies that came out that do use or leverages a combination of frameworks that when combined together really helps you to track through lesions. And these frameworks are listed here, and they allow really a more reproducible track segmentation approach, and not only within subjects, but also across subjects. It's important to have reproducibility across subjects. So based on these advances, what we propose here is to go beyond the conventional lesion load approach in MS, and I propose 3 new metrics that we evaluated in a cohort. So the first metric is basically a straight up extension of lesion load, but to the tracks space, which is called the Tractogram load. And to better understand what I mean, I prepared a small toy example here. So imagine you have lesions in a subject here in purple, and then you have your tractogram. The most naive thing to do would be to intersect this with the lesions, and you would end up with a lesion tractogram. And then to have the tractogram load would simply be the ratio of this one over this one. And when I mean the ratio here, what we use here is simply the voxelized version. So it's a binarized mask of the two, to alleviate any potential problems with streamline counts, which we can assess further. Next, there is the bundle load, which is a sub-specific case of the tractogram load, which can be applied at the local level to be more targeted to pathways in the brain, which would be relevant for a specific task. So again, here's an example here. Imagine you have a bundle like the ILF, and we have a lesion here. And if you just look at which portions of the tracks pass through that lesion, you can easily compute this ratio of lesion bundle over the entire bundle. So the blue over the green. And this gives us a nice ratio from 0 to 1. And finally, the last proposed metric is the lesionometry approach. And again, for that, I have a slide. So the most common way to apply, for example, tractometry in diffusion MRI is you have a certain metric, let's say here AFD, and you have a bundle of interest, and then you can map this metric onto the bundle and either apply along tract profiling or simply take the average of that tract, but you end up with a specific value attached to that bundle. So to perform lesionometry, it's actually quite simple. You will limit yourself to the portion of the tract that passes through a lesion, And then you can either perform along-tract profiling or take the average of that tract, again, using only that portion. So the hypothesis here is you would be more specific to the effect of that lesion down the line or along the tract, as opposed to using the entire tract to extract a certain value. So we evaluated these new metrics in 40 MS patients that were acquired on the Prisma system. We had acquired a couple of anatomical images to segment the lesions, and a simple diffusion scheme with 2 shells up to B2000. So data pre-processing included denoising. So it's nice that Yella introduced this technique before, so you all know what this is now. Then we performed motion and distortion correction. This was done using the anatomical T1 because we did not have a RefPA at this point. We then extracted tracks using TrackSeg, and these tracks were clinically relevant to the tasks that were used for these patients. You're going to see later. And then we extracted also the AFD, which we mapped onto each of these tracks. So as a first result, the first thing we wanted to look at is where are these lesions located in the brain. So the most common thing to do is to bring everything to MNI space and look at the overlap of the lesions across all subjects. And it is, at least it was reassuring to see and not surprising that most of the lesions were located in known areas that are mostly in the periventricular areas and the deep white matter. And then we also looked at, across all of our subjects, which tract had the most lesion. And it was, of course, as you can see, the genu and the splenium were the most lesion tracts. And then it goes down from there. So in terms of local modeling, just to have some visual quality assessment, you can see here that at the level of the ILF, we have 2 lesions. We have one here and one here. And I zoomed on this one, and you can see that the local modeling does quite well. You could easily track through this with any tracking algorithm. You don't see any perimeter stop in there. The coherence of the ODFs are quite well aligned. So that gives us good hope to be able to apply tracking for the next step. So here's another example of another subject where we had a lesion here in blue on the FLAIR image and a couple of other here. And then if you focus on this one, you see that the arcuate fasciculus also can track quite good through it and gives us good hope that we can sample what's happening in there first using the tracks, and also that our bundle load approach will be able to split this part of the bundle over the entire bundle, which what you would not want to see is tracks that finish here all around this. So looking at the results, first thing we want to do is compare the basic lesion volume and lesion load approach, which is what the most of the MS community is using right now, to a clinical score, which is the MSFC score, which is just a composite score of disability. And I was quite surprised, actually. You can see that the correlations are not that bad as what you would expect for something that should not correlate with disability, actually. But I suspect that it's probably due to these 2 points here. You can see these seems to be kind of outliers. So if you were to remove these 2 subjects, probably the correlation would not be as that strong. Then if you compare this to our global metric, which is the Tractogram load, you can see that the correlation is actually lower than lesion load and lesion volume, but the spread seems to be more tighter. You see a more nice spread of the values. And the other thing is the range of the Tractogram load goes from 0 to almost 100%, because as soon as a lesion is in a crossing area, you have a lot of tracts that crosses through that, as opposed to here, a simple tiny lesion, which the spread of the lesion load goes to up to 2% of the brain, basically. So that's one of the results. Next, we look at bundle-specific results. So this is the bundle load. I have 3 tasks that were administered to the subjects. And how I arrange these graphs is I want to compare basically the local bundle load. So these are all the tracks, and the global metrics, which is lesion volume, lesion load, and tractogram load. How do they correlate with the task? And what's nice to see is that, for example, in the verbal fluency task, the ILF had higher correlations than any of the global metrics, which means that, and we know that the ILF is used in, is involved in verbal semantic tasks. So it might be some sort of nice reassuring way of seeing that if you're more specific to the bundle that you're targeting with a task, you get higher correlations than global metrics like lesion volume or lesion load. And in terms of lesionometry, we did the same approach. So we compared using the entire bundle. So remember, this is the AFD average over the bundle, and correlating it with tasks. And the same approach using the lesions bundle. And now I represented the results is if this approach gave stronger correlation with the task, you're going to see more orange. And if this approach gives stronger correlation with the task, you're going to see more blue. So overall, you see their trend that most of the time the orange or the lesion approach gave stronger correlation with the task. And what's interesting again here, this is an information processing task. We know that the cingulum is involved in that task, and we almost double the association or the link with that task using the lesion portion of the bundle as opposed to simply using the whole bundle. So the key points here is that although tractogram load did not improve the strength of the correlation with lesion load, with clinical correlations, you can still see that it could be useful probably in other approaches, and it's still worth to further investigate. The bundle load, they also provided some information that would go beyond the global approach, so you're a bit more targeted to what's happening at the bundle level. And the lesionometry approach showed a stronger association with some tasks than, again, whole bundle averages. So the idea is just trying to be more specific to the lesion here. So in summary, yes, lesions are quite important in MS, but they don't explain the clinical radiological paradox as well. And we introduce a new set of tracks metrics that should be easier to use to quantify the extent of damage. And this is well in line with the disconnectome mapping approaches that's been proposed in the past. And with this, I would like to thank you for your attention.